Hello, everyone. Thank you for joining us for our April Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs at the Huntington's Disease Foundation. I want to start by thanking everyone who supports the HDF. Because of you, we are able to fund exciting science that is transforming what we know about HD. And of course, a big thanks to all of you for tuning in. Thank you for being here. We are joined today by someone who probably doesn't need an introduction to this crowd, Dr. Jeff Carroll. Most of you may know Jeff from HD Buzz, the plain language science news website that he co-founded that has become essential reading for the HD community. But Jeff also wears a lot of other hats. He's an Army veteran, a neuroscientist, an HD researcher, and someone who brings a deeply personal stake to his work as someone who carries the gene for HD. In 2025, Jeff was awarded HDF's Leslie Gehry Prize for Innovation in Science, and today he'll be sharing some of his recent work looking at a small toxic fragment of the Huntington protein and what he's learned from some recent mouse studies. Before I pass it over to Jeff, I want to remind you that you can ask questions in the Q&A box throughout the talk. So try to remember to use the Q&A box, not the chat box. We should have about 20 minutes at the end for a Q&A, so send your questions in as you think of them so that you don't forget, and we will address as many of those as we can. Also, a reminder that this will be recorded and available on our website and YouTube channel in about 2 weeks. So if you want to watch it again, or if you want to share it with someone, it will be available for that. Thank you again to everyone for being here, and I will pass it over to Jeff.
Uh, Thanks, Sarah, for that really kind introduction. It's funny. I just very coincidentally was just talking to a New York Times reporter who is doing a story about Nancy Wexler, and it reminded me, what an incredible story the Wexler family story is, and this organization is. And I can't tell people, as a scientist who's been working on HD my whole grown-up career, what an honor it was to get the call from all those folks to announce that award. It was honestly one of the highlights. You get a lot of awards in your career from organizations at different levels, but something like the HDF, really priceless to get that recognition from such an organization that's so close to my heart. I always like to start these talks with a little reminder that, although I'm a scientist now, and that's the hat I have on now, fundamentally my relationship to this is as a family member, as with so many people here. This is my mom, Cindy. This is the picture that I grabbed just because she looks so happy with four of her kids. Unfortunately, it's missing two of my siblings, so I should probably find a picture where she actually has all six of us on her lap. But it gets hard with six. So my mom, Cindy, passed away from Huntington's disease, which was, of course, something I didn't know anything about. I found out my mom was sick when I was a young soldier, actually in Kosovo on a peacekeeping mission, with no background in science or understanding of what HD was. And of course, went down the world's worst rabbit hole on the very nascent internet at the time to try to figure out what it all meant. And after I got out of the Army, I started my educational training in Vancouver, British Columbia, which was very lucky because this very amazing HD scientist, Dr. Michael Hayden, has run HD research and clinics in Vancouver that are totally top notch for many decades, and still working there today. And worked for Michael for many years. I won't say how many years it took to finish my PhD. It's not a number I'm particularly proud of, but I did a lot of work with Michael, and learned everything I know from science about him, from a very low bar to begin with. Because I think I recognized and had that experience of going from being the kind of confused guy in Kosovo trying to Google didn't exist yet, but Yahooing, like Huntington's disease, the stuff that was online was really terrible. And that was the motivation ultimately to start HD Buzz, with my work wife there, Professor Ed Wild, now at UCL. I show pictures with Ed, with my actual wife, Megan, there, who, of course, in supporting me, has also been a huge contributor to the field. And just to discriminate, my two wives, my work wife and my actual wife. And we're, I think both Ed and I are very proud that HD Buzz is still going. I mean, we both wish it wouldn't be needed, honestly. But given that it is, we're very glad that it's still happening and that Rachel and Sarah are doing such an amazing job running it. This talk is the first time I'll ever mention this publicly, but I am moving. So I have been at the University of Washington, where I run a lab in HD. And literally 2 weeks ago, I accepted a job at the Allen Institute, which is a independent nonprofit brain research institute in the Seattle area where I am. That has an absolutely amazing history of open, big science that I'm incredibly excited about, and it's not official yet. So this hold this space. This isn't officially launching until June, but since we're friends here and I'm giving this talk, I thought I would mention and put out that my email address will soon be there. Okay, so I geared this talk, I hope, as best I can for people who don't have a lot of science background. And so apologies to all the smart people on the call, including all those folks whose papers I'm about to highlight. Um, but we've known since 1993 what causes every case of Huntington's disease. That led in large part with huge contributions from Nancy and the rest of the HDF folks and organizing this consortium that ultimately cloned the gene. Was a pivotal happening in human genetics. It was insight into why people develop Huntington's, and it was an expansion of this repeat. Unlike some other diseases that are caused by a single kind of base pair mismatch or spelling mistake in the DNA, Huntington's is caused by a different type of mutation. It's still a change in the DNA, but it's an expansion of a repetitive sequence for reasons that are still being studied by scientists. Nature loves these repeat sequences in our DNA, and so it's very common in our DNA to find these long repetitive sequences of repeated letters. And in this case, the DNA letters are CAG, and they're repeated even in people who don't have Huntington's disease about 20 times or so at the beginning of what we now call the Huntington gene, since it causes Huntington's disease. And in people with Huntington's disease, those repeats are usually 40 or more. So there's a jump in the size of that repeat range. So it's not that there's a change from one DNA nucleotide to another, but rather there's an expansion of this already repetitive sequence. What does that get used for? So I almost cried when I saw this. This is you can't really see anything this small, but this is a computer representation of what one of the Huntington protein molecules would actually look like. And through these incredible new technologies, we have the ability to actually see what these things look like. So this is not the DNA. This is the protein that gets made from the DNA. This is one molecule of the Huntington protein, which again I never thought we'd live to see, but science is pretty cool. In terms of the rest of the background, so Huntington's disease is what's technically called an autosomal dominant condition. All that means is that it's not more common in men than women, although people still say that in some families, unfortunately. And that you only need to inherit one copy of the gene to have, or one copy of the mutated gene to develop symptoms. And so, there are other diseases where you need to inherit two copies, and you might be a carrier, but HD is not like that. It's quite simple in its genetics. So it's not the DNA we think that causes the problem. So the DNAs are used by cells as recipes to make these little molecular machines called a protein. And so we call the protein Huntington, which is very confusing, and we call the gene Huntington. And even more confusingly, the DNA doesn't get read directly into the protein language. Instead, there's this intermediate species that's called RNA that has the same information, but just in a different related chemical language. So you can talk about the Huntington at the DNA level, at the RNA, and at the protein level. And I wouldn't torture you about this if it wasn't relevant for the talk. This slide is not meant to be understandable. Sorry, this is just meant to be sort of a spaghetti glob of all of the different ways that people are targeting this problem. But just to say that, in general, we have this, the benefit of HD being a really horrible single mutation causing disease condition is that there's a pretty simple conceptual way to try to fix the problem, which is to get rid of the levels of the mutant gene product that cause the disease in every single case. And so this generally is called Huntington lowering. And I'll walk through this in a more simplified diagram where you can see it. But basically, just the idea of, since this mutant gene causes this mutant protein, let's get rid of some level of this cascade of events and block it and try to stop it. And I have spent a big chunk of my career on various aspects of this, starting in Michael's lab and Vermont. But there are a lot of questions about this. There's two different copies of the Huntington, the regular one and the mutated one. And should we knock down both? If we do knock them down, both of them, how low should we go? Should we target the DNA level rather than the protein level, et cetera? And so all of these are open questions. And I stole this diagram off of Wikipedia before I realized it actually has a mistake in it, which is funny, but it just shows one arm of what it's supposed to show. But anyway, this is just a cartoon of the kind of molecule that I have spent a lot of years working on. They're called antisense oligonucleotides, or ASOs. I guess in that like orange single stranded color there. And these are delivered as drugs. So they're heavily chemically modified forms of DNA that enter the cell and can change the levels or the activity of specific target RNAs. In this case, of course, the RNA that we want to go after is the Huntington one. And so the drug has been developed for many years now by Ionis and Roche is a drug of this type. So basically the goal of it is to go in there and bind and change the Huntington RNA. When I was putting this talk together, I realized I've been doing this for a very depressingly long time. I mean, it's been actually really fun, which is why I'm still doing it, but it's depressing because we're still working on this and we haven't solved this problem. But my first Huntington antisense paper in Michael Hayden's lab was in 2011, and the paper I'm going to be talking about here in a minute just came out just now in 2026 in a very recent paper from the lab. So this species of Huntington that I'm going to talk about is called Huntington 1A. And it's yet another form of Huntington that scientists have been thinking about, and that I have kind of accidentally become interested in, as you'll see during this talk. So what is it, and why do we care about it in HD? Okay, so here's the kind of stripped down version of this. So Sarah, can you tell me if you can see my pointer?
Yeah. Yes.
Okay, great. So I won't use it a ton, but just in case, especially in the webinar, people at the recording, people want to look at this. So we've got at the DNA level, we have our Huntington gene. They're in these little chunks or chapters called exons. It doesn't really matter, but they're really long. This is not really to scale. Huntington's huge, unfortunately, for those of us working on it. And there's like 67 of these things. And the one that has the CIG repeat in it is actually the first one, the first little beginning chunk of the Huntington gene. So normally, the machinery goes through, it makes this copy that I told you about called the RNA copy. Through a process in the cell called transcription. That gets sent out to the sort of factory region of the cell. And then another special machine does a process we call translation and turns it into the actual machine that we think does all the bad stuff, which is the protein. And remember, almost every HD patient has one mutated version of the gene and one regular copy of the gene from their other parent. There are very, very rare people, which actually the HDF helped find, that have both mutant copies, but it's incredibly rare. So for all intents and purposes, we can say that almost everyone has a regular version of the Huntington protein and gene and mutant form. And that's the normal process. And what was discovered actually now about 10 years ago by the Bates lab in London is that when the CAG repeats get really long, a bunch of stuff gets wonky in the cell in general. That's just true. But something in particular about this transcription process changes. And so there's a sort of an error or a shift where instead of making this whole RNA version, there's a process that gets aborted early, and you end up with this very short version of the Huntington gene that just includes the CAG repeat chunk. And that's called Huntington 1A. This is just kind of it's literally a made up name that scientists gave it because it's really hard to describe because it's not a normal form of the RNA or the protein. It's just, as I said, it's about 1/70th of the entire length of the normal Huntington RNA because it's just that first chapter or the first chunk of the coding part of the gene. Unfortunately for us, that chunk includes the CAG repeats, and those, as in the other full-length proteins cause bad stuff. This little form is translated and makes this little protein that you can imagine, as I said, is about 1/70th of the actual full-length form. So this small little tiny bit, we initially weren't sure if it really mattered. It seems to only happen when the CAG repeats get really, really long. But it is now thought by a lot of scientists who think about this, that in the brain cells of HD patients, our CAG repeats might actually continue to lengthen during age. And so even if you only inherit, as I did, 42 CAG repeats from your affected parent, there might be individual brain cells that within the vulnerable part of your brain that now have like 100 or 150. And that process is called somatic instability. It's not the focus of this talk, but suffice it to say that even in people who inherit 40 something repeats, this long repeats might eventually happen, at least in some small bits of your cell, a small set of your cells. One of the reasons this is timely, as folks in this call will probably know, that there's been this kind of exciting news from the drug company Unicure, who have had in a small gene therapy trial what looks like a really exciting potential response in a small number of HD patients, a couple dozen, but nevertheless an exciting looking response. One thing about that drug is that because of where it targets, it happens to hit both the Huntington 1A and the full-length Huntington protein. So you can imagine if Unicure's drug's coming into the cell, the place where it interacts with Huntington is here, right in the beginning of the gene. And so they've shown in non-human experiments that their drug blocks both this Huntington 1A species and the full-length Huntington. So neither they nor me have any idea whether the fact that it blocks this little funky fragment is important or relevant to the benefit they saw, but it's definitely interesting coincidence at this point that is making me and other people curious about it. So that wasn't our original question, had nothing to do with that. It was actually a very simple question, which is the kind that I like in my lab, which is that question I alluded to earlier, which is, okay, we've got two copies, a mutant and a regular one. Is it better in our mouse experiments that we can do in my lab to just get rid of the mutant Huntington protein and leave the regular one around, or does that not matter and it's just as effective to lower both copies? The reason we're asking this question is that it's actually technically much easier to hit both copies. It's much less complicated in humans, and so if it's just as good, we'd rather do that. Before I end, I wanted to highlight the fact this is really all the hands-on work of Bobby Bragg in my lab, who's worked with me on Huntington's now for 10 years or more, losing track, and it had really led this work in collaboration with Wave and myself. I'm out here talking about it, but he's the one who actually did it all. So this, going back to our diagram, this was our original very simple-minded question, not taking into account this Huntington 1A stuff at all, because we weren't thinking about it. So is it better to just do this and knock down just the mutant Huntington protein down here? Or if we knock down both of them, is that better? We worked with Wave, who are also running human Huntington's ASO trials. So they use the same kind of drug we do, the ASOs, and we worked with them and they were really good. And the mice that we use, like people, have two different versions of their Huntington gene, a regular one that's not mutated, and then one that we've fiendishly bashed in a really, really long CAG repeat that looks just like the Huntington's mutation in people. And mice never get HD, right? I will never tell you that this is a true model of Huntington's disease, and our mice have Huntington's, and if we cure these symptoms in the mice, we've cured HD. It's definitely not true. Mice are mice, humans are humans. Only humans have HD. There are many differences between mice and humans, but nevertheless, it's unethical to do these kinds of experiments in humans, and so we do them in mice. And so Wave was able, because of the way that we made these mice, to make us an ASO, which is shown here in red, that only recognizes the mutant version of the gene. And so we hope will lower levels of just the mutant gene and protein. And then they made us a matched one that targets downstream that can hit both copies. And I've tried to just call that mutant copy ASO or both copies ASO, because otherwise I get confused, and so I'm sure you will as well. So basically we had asked them, can you make us an ASO that will let us address this question? Does lowering both copies of Huntington provide more rescue than just lowering one copy? The bottom line is they were able to do this. So I've tried to simplify the cartoons here, but this is our actual data. So these are measurements that we made of Huntington, and using really cool new chemical tricks and plate experiments that we can do in the lab and high throughput. We can separately measure the levels of the regular Huntington protein and the mutant Huntington protein. And we treated mice and then measured these in their brains some weeks after we treated them, either with just a control, just a saline injection, just a saltwater injection as a reference. The ASO that targets both copies. And so we think, we'll hope will knock down both the regular Huntington and the mutant Huntington. And if you look at these graphs, you can see that's what happened when we treated the mice with the ASO that's downstream and that can hit both of them. It lowers the levels a lot by like 80% of the regular Huntington. And then you see the same deficit in the mutant Huntington. If we take that same ASO, the other ASO, the mutant only ASO, when we treat mice with that one, it doesn't change the regular Huntington levels at all. But when we look at the mutant Huntington levels, it very strongly reduces them. And I highlighted this in red just to remind myself, this is the most important feature of this data. Both of these ASOs lower the mutant Huntington, which is the bad guy for HD, to the same extent. So, okay, we've got these reagents thanks to Wei and Bobby's work. And the experiments that my lab does, we basically run like little clinical trials, but we do it in mice. We take HD mice and regular mice. We treat them both with either some control solution or a drug that we want to test its benefit or how beneficial it is. And then we age them, and then we sacrifice them, and we analyze their tissues, and we look for changes that are like what happens in the HD brain. And this is one of those, and this is just visually striking, hopefully, once I walk you through it. So the red blobs here, we're looking at with a microscope through a section of the brain. This part of the brain is the most impacted in our HD mice and also the most impacted in people, which is why we're looking at it. The red just highlights the neurons, which is one type of brain cell, and it's the type of brain cell that dies in Huntington's patients. So this specific type of brain cell in HD patients progressively dysfunctions and dies, which is why we're studying what happens in it. One of the things that happens, and if we focus on the animals, the panels here on the left, this top is just a merge. It's just to show a pretty picture. These were animals that were treated with a control, just a saline. And what you can see here in these big green blobs, these are what we call aggregates. So this is chunks of mutant Huntington that are big, huge chunks that take up a big chunk of the nucleus, which I haven't shown you here, but they shouldn't be there. If you do this stain with mutant Huntington in regular mouse, you will never ever see these big clumps. They're very specific, and they also happen in HD patients. There's been a lot of debate about their exact role, but they're definitely like a bad thing in the brain that happens in HD mice and HD patients. And you can see that they're very abundant. 30 or 40% of the neurons in this brain have these aggregates. When we treat with the ASO that targets both the regular and the mutant Huntington, we actually see no benefit. There's a bunch of these aggregates. I took out the quantification because I don't want to overwhelm people, but this effectively didn't change the amount of these at all, which is a little bit surprising because we did lower the levels of mutant Huntington pretty substantially in these animals. On the other hand, when we treat it with the ASO that targets just the mutant one, there's almost none of these. We left one in just because we don't want people to think we're lying, but they're more or less gone. There's another technique that I won't even describe, but I'm happy to talk with people about this afterwards, called RNA sequencing. It's a way of, instead of measuring one thing like the Huntington gene, it's a way of measuring every single transcript in the cell. So every gene's activity in the same cell, and asking, what are those levels in these two different conditions? In this case, of course, in HD mice that are treated with our two different ASOs. And when we looked at, and we measured, like 25,000 different genes all in this one experiment, which is why we like it, because it's a little bit less biased. And what you can see is that we actually only, by treating with the ASO that targets both Huntington copies, there's almost no changes. We only change Huntington, which, thank God, or this would have been a horribly failed experiment. We know that we knocked out Huntington. That's what our ASO does. But if we look at the effect of the ASO that targets both wild type and mutant Huntington, you can see this big shift of both up and down of all kinds of different genes activities. That could be good or bad. What we showed by carefully looking at this is that it was really good and that it was providing a lot of rescue. So this was very striking. Again, with the ASO that we know lowers Huntington, we did not see much rescue unless we targeted the mutant only one. So I was just showing this work to Jill Bates, who's a professor at UCL in London, as we were talking through our work together. And she pointed out to me that, her lab had discovered this funky Huntington 1A species. And because of the way it gets made, she thought where our ASO was, it might also hit this Huntington 1A species. So I'll walk you through some data from our paper that we generated to answer her question. So this is a little confusing. So I'll walk you through. This is like a brand new fancy DNA sequencing technique that we can actually count the numbers of this Huntington 1A species and differentiate it from regular Huntington, which is otherwise pretty tricky. So what you're looking at here is a cartoon of the Huntington gene. So this is where this Huntington 1A species, this little variant gets made. And what you're looking at is information from 4 different control mice, which are these 4 different green lines, 3 different mice treated with the ASO that targets both copies, or again, 4 more mice that were treated with just the mutant ASO. And so I'm showing you individual animals so that you can see the raw data. And this is literally the raw data. So what we've done here is we've counted every time that we read a species of this Huntington 1A, we make a dot, and then those dots eventually pile up into this kind of little like mountain landscape. And so you can see basically there's a lot of this Huntington 1A counts in these animals and these animals, but almost none in these. So this is literally counting the number of the Huntington 1A species and doing a little histogram to show like how many of them are there piling up there. And you can just visually, hopefully, tell consistently across these animals that only this ASO leads to that reduction. So Jill was, of course, right. What was cool and unbeknownst to us when we started these experiments is that Jill and Anastasia were doing a parallel experiment to ours using a different chemistry that has a different type of benefit and disadvantages, as with any kind of approach in the lab. And so, this often happens in science that kind of people have a similar idea at the same time because there's kind of the question that the field kind of needs to address in the moment. And this was one of those happy accidents that Jill and Anastasia were doing this with a different chemistry, but like overlapping benefits and disadvantages at the same time as us. So ultimately, we've been working together throughout this, and Jill's on her paper and stuff, and we published these just a couple weeks ago in a journal called Science Translational Medicine as parallel papers. So uh, I made this cartoon not originally for this audience, but for scientists and clinicians who I gave this talk to, because this is confusing even to them. So don't feel bad if the details of this are a little too much for this one webinar. But let's just say that there's different types or different approaches or different chemistries that people can use to do the same thing. And so, we use antisense oligonucleotides just because we have a lot of experience with it and we've been doing it for a long time. Those work in the cell in a certain way. And the way that works, it's technically impossible for us to directly target Huntington 1A itself. Because that's what you would want to do, right? Like, okay, if you think this Huntington 1A thing is a big deal, then make an ASO that only hits Huntington 1A. But for reasons that are way beyond the scope of this webinar, that's not possible. The approach used by Jill and Anastasia's labs is called siRNA, and it works just through it's very related but slightly different chemistry that is an RNA rather than a DNA approach. And just because of the way it works in the cells, it's able to directly feasibly target Huntington 1A itself, just by virtue of how it works. And so you could imagine they were able to make 2 different molecules of their preferred chemical approach, these siRNA molecules, one that targets just the Huntington 1A and one that targets the full length only and spares the Huntington 1A. So they were able to do the thing that we couldn't, which is to make a molecule that only knocks down the Huntington 1A species, and they could compare it to one that only targets the full-length Huntington protein, and to have that very clean contrast, the ASOs wouldn't allow us to do. And so they took a whole bunch of mice. They sort of do experiments more or less like what we do, which is to say, take a bunch of mice. They use a slightly different version, but very similar conceptual version of an HD mouse model. They treated it with either their compounds that reduce Huntington 1A in a targeted way, or only the full-length Huntington and spare the Huntington 1A species. This is just one of the many endpoints that they showed in their paper. This is a different version. This is their stain. A lot of science, like, it sounds fancy, but at the end of the day, you're counting dots on a microscope to like come up with these numbers down here. It literally is that simple sometimes. This is a different part of the brain, if you noticed, for the aficionados, but a very similar stain where they're looking at these chunky aggregates of Huntington that accumulate over time in HD patients and in HD mouse models. And these are the mice that they've treated either with, in their case, two different kinds of control. So this is kind of the levels of how much aggregates happen in this animal at this time. When they treat with the ASO that lowers the full-length Huntington very effectively, in this experiment, it didn't have any impact on the amount of aggregates. And when they treated, on the other hand, with their molecule that only targets Huntington 1A, actually, even though it was much less strong in terms of how much percentage it reduced it, they actually saw a really dramatic benefit to knocking down just that Huntington 1A species in this animal model. Like us, they did this RNA-seq, this kind of like unbiased snapshot of the levels and therefore activity, presumably, of all the different genes in the genome, as opposed to just measuring one or two at a time. And as with us, actually, they did see a little bit of rescue of this pattern of altered gene expression that happens in their HD mice when they treated with the molecule that hits Huntington, the full-length Huntington. But when they did their intervention that blocks Huntington 1A directly, they saw a much better rescue of this unbiased signature. And this is striking for a couple of reasons. One, because actually that drug was just less potent just by virtue of how it was screened, and so they were better at knocking down full length than Huntington 1A, and nevertheless they saw this benefit. And also in this treatment, they, unlike us, whereas we don't have this ability with our ASOs, they were able to completely spare full-length mutant Huntington in these treated animals. And so it seems that knocking Huntington 1A, at least in this experiment, was actually more effective than was lowering the whole Huntington gene itself. And so I know there's going to be a lot of questions. So I wanted to kind of end with this summary, coming back to this sort of overlay of how cells take on ASOs and these other siRNA molecules and where they work and what they mean. And just to say that, like, both of these experiments suggest that Huntington 1A targeting was required to get the maximal benefit of these experiments. And the final thing I want to say about our results is that I bolded it here in mice. It's very important from my point of view to remember that these are mouse experiments, and we use mice because it would be unethical to test early hypotheses and unregulated drugs like we're doing here in humans. And I wouldn't want to be the human that got tested in. So they're kind of our least worst solution. But there are a lot of differences between our HD mouse models and HD patients. And one of them that's worth saying here is that when the Huntington gene was first discovered, people started making mice that had CAG repeats that were the same length as humans, like 45 or 50 who have HD. And a very striking thing about those animals is there's absolutely nothing wrong with them. So the same CAG repeat that causes a horrible disease in human does absolutely nothing to a mouse for reasons that we don't understand whatsoever. And so one of the kind of dirty tricks of our, people who do HD mouse models like me is that the mouse models we use have really, really long CAG repeats. The ones we used have 111 CAGs. The one that Jill's lab uses actually have 175, which is like longer than almost any human patient ever. And it turns out that you need those long CAG repeats to drive these, the changes in these mice, like these aggregates and these gene expression changes that we use to test these things. And it means that those things develop over time in animals in a lifespan that's consistent with an experimenter life. But it's not what happens in human patients who, of course, have much, much lower CAG repeats. And so I just want to highlight that for many reasons, these animals are useful, but they're also very unlike humans, and so I would have absolutely no um, concern with myself or one of my family members recommending to them to take a drug that does not target Huntington 1A. I'm not saying that we should stop the presses and not do this. It's just that these mouse experiments suggest some new understanding or gaps in our understanding, which is the extent to which this Huntington 1A species is important. And we want to use these mouse models to propose that people working in clinical trials think about this. And for example, design tools to measure the Huntington 1A species itself, which is very tricky currently, to say the least. And start paying attention to it and start building up a better sense in humans where it matters if this Huntington 1A species is important. So I'm really jazzed about this. With my scientist hat on, it's super cool biology. This question of like, why is this happening at all, and why is this gene that has these long repetitive regions, why does it lead to this kind of like stutter and stop where suddenly this premature form of Huntington gets made? These are really interesting science questions. I would say that in terms of as an HD family member, they're kind of like cool, nice to understand the impact of, but not something that would change my plans about which drug trial I would myself or recommend to my family to be in. And this is the first time I've ever talked about this to a lay audience. So I hope that was somewhat understandable and completely understand if I totally messed that up, and it wasn't. I really hope not. So this work was done, as I said, really led by Bobby Bragg, and a lot of folks in my lab worked hard on this, and so thanks to them, really amazing collaborators. HD, as folks might know, is an incredible collaborative research community. Jill and Anastasia and I were sharing results and talking about this early before stuff was published, which is not usually how people do this, which is how these things can happen. And we can work together to do the best version of this, because ultimately everybody's, working towards the same goal, which is to try to hasten treatments for HD patients. And I want to thank in particular the people at Wave who worked for me, especially Jing Lu, who worked on this for a really long time in the early phase to get these ASOs so dialed in to let us address this question. And with that, I hope I left a reasonable amount of time for questions, and I'm happy to take whatever questions you have.
Thanks so much, Jeff. Yes, we have a lot of questions. So the first one, someone's asking if Votoplam and Skyhawk are not exon 1 targeting, how can they bring any benefit to the disease?
Yeah, I think that's a great question. And I'll just say for people who don't know, these are small molecule drugs, which is to say like pills, like aspirin that you could take that have this incredible effect of lowering levels of the mutant RNA and protein. It's a little in the weeds, but where they target on the gene is way downstream from this Huntington 1 area. And so it's not definitive as far as I know, but I don't expect them to knock down this Huntington 1A species. All the preclinical data, meaning all the experiments they've done in monkeys and mice, all the stuff they've done suggests that it is beneficial. And so I think that highlights that what this is telling us in these mouse experiments is like at the margin of these super long repeats that we use in mice, there might be a special benefit of Huntington 1A, but I don't have any concern. I hope my family can take one of those compounds. I'd love to be in that trial myself. I literally am going to go to a screening visit at the Novartis thing in Seattle, I hope. So I just want to reiterate to everyone that I'm very happy those things are going forward, and I expect them to have a benefit.
Yeah, they're super exciting. It's a pill. Kind of related to that, do you think that they could develop a small molecule that's capable of specifically targeting 1A?
We've talked to people. I mean, officially, no one knows because you don't know until you try the chemistry of it. And we've talked to the people at both of those companies and another one over the years who have that chemistry that can make the pill molecules. It seems daunting because the way that Huntington 1A is made is different than most genes are spliced together. And so it seems like a very, very daunting challenge. I'm not holding my breath in our lifetime, frankly.
I mean, I think the fact that we even have a pill that lowers Huntington is mind-blowing, right?
Unbelievable. Yeah.
No one thought we would be there. Okay, so here's one. How much lowering of Huntington 1A do you think is necessary for a clinical benefit?
It's great. Great question. Don't know. All we can say, and we don't even have, neither Jill nor I have a dose response, meaning that like give a little bit of the drug and see, so we don't even have any sense from our animal experiments the dose relationship of like how much lowering is needed to have these benefits. So I would say that, when you do these mouse experiments, you try to like maximize your dose to kind of like get the kind of edge case, and that's what we did. And so I would say that we just don't know.
Cool. Here's one. You clearly see molecular changes with the ASOs targeting Huntington 1A. Do you see any functional changes in the mouse models, changes as the neurological phenotypes?
Yeah, it's a great question. Ultimately, at the end of the day, HD symptoms are not, aggregates and gene expression changes. It's symptoms, right? These mouse models that we used, they're so subtle that they basically, at the age we use them, they're essentially indistinguishable from regular mice. They don't have a neurological phenotype. We're using them specifically because we want to look at these molecular changes and try to understand the causal relationship between these molecular changes. Like that's why we're doing these experiments. So we're using the mouse model as more of a molecular model, not a behavioral model. So in our case, we haven't done any behavior at all. It's not. I mean, we could, but we'd have to wait another like year for the mice, and that's just not how we set up the experiment, unfortunately.
Related to that, someone's asking about somatic expansion. Have you looked at instability in these mice?
Um, I'm trying to remember if we actually looked at somatic instability. Gosh, I feel like, oh yes, we did. It's not changed.
Interesting. Okay.
Yeah, we've compared a lot of different treatments that bind to the DNA, like zinc finger transcriptional repressors, block somatic instability. But things that target the RNA, like these ASOs, do not generally in our experience.
Okay. So there are a few questions about aggregates. So this person asks, are the aggregates the problem or just a product of the disease? Would a drug that only clears aggregates work?
I love that question. I don't know the answer to it. It's the field, I would say, has changed a lot. When these aggregates were first found, and just to say that like aggregates in Huntington are very similar to like plaques and tangles and Alzheimer's, and so all of these diseases seem to be associated with these plaques or tangles or chunks of crap, basically, that accumulate in the brain. And in Huntington, they happen to be these aggregates. And so, people discovered them. They thought, oh, they must be the cause of the disease. And then there became some kind of controversy and confusing data about, how important they were. I sort of use it as a kind of a marker of stress, right? Like they shouldn't be happening, and they correlate with what happens in humans. And in our case, they correlate with the gene expression changes in a very compelling way. So for me, they're a piece of molecular evidence, but not something that I would say is like, I think therefore we've cured the disease. I love the. I've been chatting with this guy David Baker here at UW, who is won the Nobel Prize for doing. They can make a protein, and one of the things we've asked them is to make us something that can just clear up aggregates, and like just only do that, and then would we rescue the disease? So that's something we're very interested in as well.
That's super cool. Uh,
Hmm.
So here's someone who's asking about delivery. What kind of drug delivery can currently lower Huntington 1A? So maybe you could talk about how the ASOs and siRNA is delivered.
Yeah, so both ASOs and siRNAs, these two different types of therapies that Jill and I used in our mouse experiments, are used widely in humans, in Huntington's and outside of it. They're all both delivered intrathecally, which is to say in the spinal fluid. So they're a small injection made at the base. They can last a surprisingly long amount of time. And just to say, Alnylam Therapeutics has a siRNA, which is like the chemistry that Jill Anastasia used, program for Huntington's. There's targets exon 1. And so we don't have, as far as I know, there's not data, but I would suspect that they lower Huntington 1A levels with that siRNA drug. So it is possible to do it. You just have to target the right part of it. And luckily, Alnylam has with their new program.
Thank you. Sorry, some of these questions are long, so I feel like I'm not really
It's okay.
absorbing what you're saying. So here's
You
someone
tune
who's
me out, Sarah. It's fine.
I do it all the time, Jeff. So here's someone who's asking maybe about the difference between juvenile HD and adult onset. They want to know if the aggregates contribute in any way to JHD.
Yeah, as far as we know, like, biologically, there's not really much difference between JHD and HD as far as we know. It's the mutation. it's just that in JHD, that expansion process has gotten there earlier. So I don't want to claim this confidently. I don't actually know of aggregates in JHD. Can you picture any data in your head, Sarah? Like,
Um, I mean, it's
yeah,
so phenotypic forward that people usually talk about that over, and we can't
yeah,
really model it, right? Like this isn't something we're technically everything we're modeling is JHD.
yeah, exactly. All of our mouse models are actually JHD models, to be honest.
Mm.
Um, yeah, so I don't expect that these processes would be different or that therapies would work differentially better in JHD that target all of this. I think JHD would be just as eligible as HD.
Thanks. So here's a question about the transcriptomic data you showed. They say, if I understand correctly, Huntington 1A only knockdown showed much improved recovery in terms of transcriptomic than full-length Huntington only knockdown. Do they share the recovered gene list or are they quite different? If we target both mutant full-length Huntington and Huntington 1A, could they give better transcriptome recovery?
Definitely, yes. Uh, and I guess knocking down as many of these bad things as you can is in general the better idea. Deep thoughts with Dr. Carroll. Knock down all the bad stuff. That's better. Uh, yeah, but I just a minor correction. We don't actually see any rescue in our full length Huntington knockdown, which is striking. There's literally two genes that are DEGs, one of which is Huntington. So, we measured 30,000 genes, or not 30,000 genes, 20,000 genes or whatever, and uh, only two of them are changed. One of them is Huntington because we knocked it down. The other one is an immune gene that's upregulated, which is really interesting, but totally. Yeah, so it's not that we have modest rescue. That's kind of more what Jill saw in her experiment. We don't see any rescue with the full length knockdown.
So this is someone who is curious about the quote unquote normal function of Huntington. Can you talk a little bit about everything that it does?
Yeah, we don't know everything that does. It really irritates me. But another chunk of my lab, that's what we work on, and we've got a bunch of papers recently about that question. Particularly again in mice where we can ethically knock it out. So we can, in mice we can completely excise their Huntington gene, their regular Huntington gene, not their mutated one that we made to make them like HD. And my lab has been working on that for a long time because that question irritates me. We discovered this thing in 1993, and we don't know exactly what it does and what all of its functions are. That was actually, that was the motivation of our original study, was that we wanted to have very clean mouse experiment where we can control all the variables to address the question of is preserving wild type Huntington better. That's the experiment we set out to do. And as often happens in science, like the result we got kind of put us down a rabbit hole that was not what we set out to address. But that was our actual goal of all of this, really, initially.
I mean, it's part of what makes it so complicated, right? It's so big, it does so much stuff. And so there might be different components doing different things. And maybe this speaks to some of your results, right? Like maybe Huntington 1A is heavily involved in aggregates, but not necessarily somatic expansion. So we really, I
Right.
think the future is moving us toward these cocktails of drugs that might be effective.
That's what I
Um,
expect.
so here's someone asking about Vico's drug. Does VO659 affect Huntington 1A?
Oh, I love these questions. These are really good questions. We don't know. We've tried to measure it, but it in a separate mouse experiment. I don't want to speak about because it's kind of their data. Uh, for technical reasons, we failed in measuring that, but we are very curious about that. So Vico has an ASO. The ASO they're testing in HD patients, actually, and other CAG repeat disorder patients. Currently targets exon 1, and so it might, in theory, block Huntington 1A formation. We tried to measure that, but there's technical reasons that I can talk to people about offline about how their ASO works that makes that very, very challenging technically. So we wondered the same thing. It might very well. And so, in addition to the Unicure that targets exon 1, the ZFP by Takeda targets exon 1, and definitely reduces Huntington 1A. We've shown that in another paper. And the Alnylam compound, I would be so those are the ones that I would be excited about if I was worried about this in particular.
So, thank you. Here's someone who's asking about the AMT 130 stuff. Given what you know publicly and what's been going on with the FDA, from your experience, do you think that the drug is working? Obviously, it's only been 3 years, but it seems like the placebo wouldn't last that long. They would love to hear your thoughts on that topic.
Yeah, I'm a layperson here. I don't have any professional data from them. But the data I've seen is very compelling. Particularly for me, there's a measurement that you can do of a protein called neurofilament light, or NFL. I think HDF has another webinar about that. And it's basically a marker of the stress of neurons. And in HD patients, of course, they have lots of stress neurons. It goes up, very, very reproducibly over time. And it's a very objective, it's a lab measurement. It's not like watching somebody walk or whatever. All respect to neurologists, uh, it's a very objective lab measure, and in HD patients, it goes up full stop, right? And in the small number of uniQure patients that got to that 3-year time point, it was below baseline, which to me is very, very striking. And it means there must be something going on. Now, the magnitude of the clinical benefit is not my pay grade. I'm not an MD. I don't know that stuff. And the specifics about the control group and whether it's the appropriate one or way outside my pay grade. But I find it very compelling. And it generally, if nothing else, it says that Huntington lowering, which seems like a really good idea, and I've been working on it for my whole life. Uh, Went through a period where the tominersen and wave trials failed in the midst of the pandemic, and I thought, man, maybe I've wasted my life, and lowering Huntington is a bad idea. But that was the first thing where I was like, okay, cool. Like, I think maybe we're going the right way, and that's how I take it. I don't think they're going to be able to deploy on a scale they'll need to if it works. Frankly, just because it's a 17-hour surgery. So certainly the people in Venezuela and Colombia aren't going to be getting it. So we got to keep trying to find a way to do that with a chemistry that's deployable.
Yeah, and I think the fact, right, that we've had multiple trials now that are Huntington lowering, different modalities, totally different methods, and they're showing benefit. We have WAVE showing benefit. We're seeing some benefit from Votoplam. So I think overall, as an approach, if it were, one trial worked and two didn't, that might be telling. But if all three of them are showing there might be some benefit, it's definitely worth chasing, right? Like it's good that we have three showing it.
Yeah, and I think the bottom line is we're doing it. All these trials are getting run. Nobody's stopping. All these things are. I mean, I don't know what the hell the FDA is doing. If somebody does know, they can tell me. That'd be great. But like in general, all these companies are full speed ahead. The patient community is full speed ahead. We're running these trials. We're gonna know the answer.
Yeah, that's a good point. So here's someone asking about kind of the nuance between ASOs and siRNA. They say, are there advantages to the ASO approach in targeting nuclear RNA over siRNA approach that can only target cytoplasmic RNA?
Yeah, it's a great question. And not to get too far in the weeds, but there are benefits because there are like, so for example, with Wave's approach, they're targeting little genetic variants that are linked to the CAG repeat but aren't the CAG repeat itself. Almost all of those are in a region of the gene called introns that get spliced out, and so you can't target them in the cytoplasm. And so there are technical reasons why ASOs might be have benefits there. In balance, in general, siRNA is more potent than ASOs. So in perfect head-to-head comparison, siRNA is going to be more potent. And it used to be that siRNA was impossible to deliver, but Alnylam really seems to be cracking that in the brain. And so hopefully we're in an era where we can use siRNA, but there's definitely going to be times when ASO is the right approach. And for example, if you want to target one of those sequences that's not present in the cytoplasm, like in the introns.
So we're kind of coming up to the end now, so I might end on this question, and it's one that we get, I think, every single talk every HD researcher gets. But this is someone sharing their story. Their stepdaughter's father has HD. He has two kids, and they both have kids that are at risk. And so they're asking, what is the 10-year, 20-year prognosis, either for a strong treatment or a cure?
I don't use the C word. I'd be perfectly happy with getting a little bit of shakiness in my 80s or whatever. That's my goal. And whether that would match what someone calls a cure, I don't know. But I'm very optimistic about this current round of trials. You can do the math. There's phase 3 trials, which is the last stage of trials that are running or just starting for the Alnylam drug. Sorry, not phase 3 for the Alnylam, but Alnylam is starting. There's phase 3 ASO trials happening. The small molecules are in phase 3. It's very reasonable that in the next couple of years, we're going to know the answers to those. If the answers are, yes, these drugs worked, then that's your answer. And a couple of years from now, we're going to know that. It's most likely it's not going to be 100% rescue. And so the question is, how much is it? And then we have to combine these things, right? And people should take solace from things like cancer and HIV, right? In my lifetime, HIV was a death sentence. everyone who got it died. And then there was drugs that worked that kept people alive, but they were horrible. And then there was other ones that were like less horrible slightly, and they had overlapping symptoms. And so like, and now people live with HIV and have totally normal lifespans because they combine these things, they tweak the doses, they refine them, and it's a combination therapy. I think that's what we'll have for HD. But I think the first of those arrows in the quiver is going to be here sooner than later if this current generation of Huntington lowering trials works.
I agree wholeheartedly. We're so close, and it's going to be iterative.
Yeah.
Thank you so much, Jeff. So we are coming up on 1 o'clock, so I'm going to wrap it up there. We have a lot of questions that we didn't get to. So if you left your contact information, I will send those over to Jeff so he can follow up with you if you left that information. I want to end by, again, thanking everyone who donates to the HDF to support the research that we fund. And of course, everyone who tunes in. Thanks so much for joining us. And last, but certainly not least, Jeff, thank you so much for telling us about your work. Clearly, a lot of people are very interested to hear about this. So thanks for joining us.
Yeah, it's my pleasure and an honor to be here. So thank you very much.
Our next webinar is Wednesday, May 27th. We will be joined by Dr. Dirk Keen and Dr. Dan Child, who will share about the importance of brain donation and the brain bank that they have created at the University of Washington. Registration for that webinar is now open, so you can head over to the HDF website right now and get it on your calendars. We are so proud to showcase the advancements in HD research through these Research Spotlight webinars. If you find these sessions valuable and you want to support our mission, please consider donating. Thank you for being a part of our community, and we hope we will see you next month.
Thanks, everybody.